Good morning, it's July 28th, and this is your daily brief in science. Here's everything you need to know. A recent study published in Science Advances has revealed astonishing findings about life beneath the Earth's surface. Researchers have discovered that the deep subsurface biosphere, once thought to be uninhabitable due to the absence of sunlight and nutrients, is actually home to a diverse array of microbial life. The study highlights that these microorganisms rely on inorganic redox reactions for energy, primarily utilizing hydrogen produced from the interaction between water and rock. By simulating crustal faulting, researchers demonstrated that the fracturing of rocks generates free radicals that decompose water, resulting in energy-rich hydrogen and oxidants like hydrogen peroxide, which are essential for microbial metabolism. Notably, the research found that hydrogen production from earthquake-related faulting can be up to 100,000 times greater than from other known sources. This significant output influences the redox cycle of iron and other elements, sustaining microbial life in the deep biosphere while also driving the geochemical cycles of carbon, nitrogen, and sulfur. These findings challenge the long-held belief that all life depends on sunlight, revealing that microorganisms can thrive in extreme environments using energy derived from chemical processes. The study also suggests that fracture systems on Earth-like planets could potentially provide habitable conditions for extraterrestrial life. Led by Professors Hongping, He and Jianxi Zhu from the Guangzhou Institute of Geochemistry, this research was supported by various funding sources, including the National Science Fund for Distinguished Young Scholars. The implications of this study are profound, as it redefines our understanding of life in extreme conditions and expands the search for life beyond our planet. A breakthrough study from UC San Francisco has pinpointed a specific receptor, ADG-RG1, as vital for microglia in their fight against toxic amyloid-beta plaques linked to Alzheimer's disease. This receptor is essential for enabling microglia to engulf and break down amyloid-beta, a protein associated with the progression of Alzheimer's. The research indicates that ADG-RG1 enhances microglial activity, which is crucial for maintaining brain health. Without this receptor, microglia struggle to digest amyloid beta effectively, leading to increased plaque formation and neurodegeneration. Notably, individuals suffering from severe Alzheimer's showed significantly low levels of ADG-RG1 in their microglia, correlating with a substantial buildup of amyloid plaques. In contrast, those with mild Alzheimer's exhibited higher levels of ADG-RG1, suggesting that their microglia were more efficient at preventing disease progression. Experimental results from mice lacking the ADG-RG1 receptor demonstrated rapid plaque accumulation and marked memory decline. Conversely, humans with elevated ADG-RG1 levels displayed milder symptoms and fewer plaques. The discovery of ADG-RG1 opens new avenues for potential therapies aimed at enhancing microglial function in combating Alzheimer's disease. This research emphasizes the possibility of targeted treatments that could boost the brain's immune response to amyloid beta, paving the way for more effective Alzheimer's therapies. ADG-RG1 is categorized as a G-protein-coupled receptor, presenting opportunities for developing new drug therapies focused on this receptor to improve outcomes for Alzheimer's patients. The study received backing from notable organizations, including the National Institutes of Health and the Alzheimer's Association, underscoring its importance in the ongoing research into Alzheimer's. MET functions as a receptor tyrosine kinase that activates signaling pathways crucial for cell proliferation and survival, making it a key target in cancer therapy. In non-small cell lung cancer, or NSCLC, mutations in MET exon 14 serve as significant oncogenic drivers, leading to sustained receptor activation. This has paved the way for the development of targeted therapies, including MET tyrosine kinase inhibitors. However, MET alterations extend beyond exon 14 mutations. They include gene amplification and protein overexpression, which play a vital role in the progression of various solid tumors, influencing tumor initiation, invasion, and metastasis. These alterations are not limited to NSCLC. They also appear in other cancers such as gastric, colorectal, and hepatocellular carcinoma, indicating that MET-targeted therapies could potentially benefit a wider patient population. Accurate diagnosis of MET alterations is essential for effective patient selection. Techniques like next-generation sequencing and immunohistochemistry are utilized to identify actionable mutations. The increasing understanding of MET biology, coupled with the emergence of novel therapeutics, signifies a shift towards personalized cancer treatment strategies aimed at maximizing patient benefits while minimizing toxicity. The treatment landscape for MET-altered cancers is evolving, moving from traditional tyrosine kinase inhibitors to innovative therapies, including anti-MET monoclonal antibodies and MET-directed antibody drug conjugates. A significant advancement occurred in May 2025 when the FDA approved telisotuzumab vedotin, a MET-directed ADC for advanced-stage non-squamous NSCLC with high MET expression. Despite these advancements, resistance mechanisms to MET-targeted therapies, such as secondary mutations or activation of alternative pathways, highlight the need for combination strategies to enhance treatment efficacy. Adverse event profiles can vary among MET-directed therapies. Common toxicities associated with TKYs include peripheral edema and nausea, while ADCS may lead to hematologic suppression and neuropathy. Additionally, the interplay between MET signaling and the tumor microenvironment affects tumor progression and responses to therapies, suggesting that combined therapeutic approaches may improve overall efficacy. In summary, while MET alterations complicate patient treatment stratification, they also indicate potential sensitivity to MET-targeted therapies, paving the way for more effective cancer management. Researchers are making strides in the fight against aggressive cancers, with a breakthrough method that could change treatment protocols for melanoma and triple-negative breast cancer. Fleury-Augustin Nsole-Bitege, a researcher at Northeastern University, has developed a technique that utilizes a light-sensitive chemotherapy drug to target cancer cells more effectively while minimizing harmful side effects. This innovative approach involves the attachment of the chemotherapy drug to a protein called MTF, which is found in both melanoma and triple-negative breast cancer. By activating this drug with near-infrared light, it selectively targets and eliminates cancer cells without affecting surrounding healthy tissues. When the localized light is applied, it triggers the drug bound to MTF, leading to the production of cytotoxic reactive oxygen species that cause tumor cell death. This method is particularly significant for treating triple-negative breast cancer, a type that is notoriously difficult to manage due to the absence of 3 critical receptors, rendering standard therapies less effective. Patients with triple-negative breast cancer often have to rely on surgery, radiotherapy, and traditional chemotherapy, which can lead to severe side effects such as hair loss and fatigue. Nsole-Bitege's light therapy offers a more targeted alternative, potentially reducing these adverse effects associated with conventional chemotherapy infusions. His findings, published in the journal Cancer Medicine, introduce a technique known as photoimmunotherapy, which aims to deliver chemotherapy directly to cancer cells while sparing healthy tissues. This research represents a promising advancement in cancer treatment, providing hope for patients facing these challenging diagnoses. A tiny meteorite named Northwest Africa 12,264 is reshaping our understanding of how planets in our solar system formed. Researchers have analyzed its composition, revealing a unique chromium and oxygen ratio that indicates its origin in the outer solar system. Isotopic dating shows that this meteorite formed approximately 4.564 billion years ago, challenging existing beliefs about the timeline of planetary formation. Traditionally, it was thought that the inner rocky planets, like Earth, formed around 4.566 billion years ago, while gas giants and outer icy bodies developed slightly later, around 4.563 billion years ago. However, this discovery suggests that the formation of rocky planets beyond Jupiter occurred concurrently with the inner planets, indicating that the outer solar system bodies were not delayed due to colder temperatures and higher ice content as previously believed. The meteorite weighs just 50 grams and indicates that rocky planets and icy bodies formed simultaneously, which could significantly alter our understanding of planetary formation. The research detailing these findings was published on July 4, 2025, in the journal Communications Earth and Environment. This new understanding not only reshapes theories about the solar system, but could also impact our knowledge of other planetary systems across the universe. The implications of these findings are vast, suggesting a more complex and intertwined formation process than previously thought. This has been your daily brief in science. To read more about these stories, follow the links in the episode bio. You can also subscribe to these updates via email at www.brief.news. For more daily podcasts about the topics you love, visit brief.news/podcasts. Tune in tomorrow. We'll be back with everything you need to know.